Now, digging in and getting really serious about looking at microbes or the microbiome in TB research. This is a publication that was published, I think it's been like almost 10 years ago, and somebody did a 16S sequencing experiment, okay? 16S meaning that you essentially go into the sample, you extract DNA, you amplify the 16S gene, and then you profile the content or the composition of 16S genes in that sample. And looking at this data set, what you're viewing here in this stacked bar chart is that every bar represents a different sample, and then the colors represent the composition or the proportions of specific microbes. So, for example, the gray here represents Streptococcus species. So again, you look at this first sample, and clearly there's a lot of strep in that sample, for example. So that's what you're seeing down here. Whereas, for example, these kind of pinkish colors are staph. And so then you can actually go through and look. Now, this study, what it did is it took sputum samples from individuals that were TB positive. It took oral samples from people with TB and control samples, and also nasal samples from TB and control as well. And then they profiled using 16S sequencing, they profiled the microbiome. And looking at these samples, you can tell there's a very stark difference, right? If you'll notice that in both sputum and oral, it turns out that the oral TB and sputum TB were looking similarly, and that makes sense, right? Because of contamination and other issues there, and proximity. If you'll notice, you're seeing a lot more strep in the TB samples than you do in the controls. And if you notice those controls, you're seeing a lot more microbial diversity. And then similarly for the nasal microbiome, you'll notice that there's high variability here. Maybe there's not a lot of consistency in what you're seeing, but you're seeing a lot more strep and overall a lot less staph. Okay, it looks like the nasal microbiome is mostly staph, and this is Cutibacterium in the red. And that staph and that Cutibacterium kind of goes away. And what you're seeing here, this green is more Moraxella. You're seeing more strep. And then some samples, they don't completely lose their staph, but the other ones do. Okay, does that make sense? So that's actually a stark difference, actually, right? You can actually very clearly see which ones are TB and which ones are controls, okay, in this context. Now, these are healthy controls, not pneumonia controls. And in fact, the pneumonia controls might be confusing here, right? Because it turns out we're seeing a lot of strep in the TB samples. So it's quite interesting. Okay, this is just kind of to whet your appetite to say that maybe there is something going on, that we can actually look at the microbiome and try to understand both what happens to the microbiome because somebody has TB, but then also, are these interacting? Okay, because the question there, thought would be is yes, that seems like there's going to be some interaction here. Yes, question.
Get your point, but this is an N of 6. Right?
Yeah, very small sample size. So we should do more study, right? Okay, I like that. Hang on to that. We're gonna show you some more data. But all of the data examples that we're using are kind of small sample size, right? So there's a lot of work that needs to be done here. I'm also showing you this one because it's the cleanest data set I can find and in this space as well. Okay. All right. So just stepping back, we're going to go data science-y computationally on you here. This is I think a very messy and possibly confusing representation of existing tools for studying the microbiome. Okay. And so, for example, people do, when you're looking at the microbiome, you want to say, okay, well, are we going to do whole genome or whole transcriptome sequencing? Are we just going to look at the 16S? That's what's over here. And then there are a whole bunch of different tools and methods and analytical approaches for analyzing these types of data. Okay, so for example, there's this method, and you can see it's kind of small here. Going back here, is this QIIME2 method is the most common method for analyzing 16S-based sequencing data, okay? This icon stands for a tool called Mother. It turns out, as true with bioinformatics, you have to name your tool something really, really cool. And anyways, and then my group, we've developed this tool called Pathoscope, and our newest version is called Metascope. But if you'll notice that there's a lot of different designs for your study. And then the problem is there's a lot of tools for analyzing those data sets as well, okay? And it turns out, I'm going to show you that especially when it comes to analyzing the 16S data, there's actually still a lot of work that needs to be done in order to analyze these data properly and effectively, okay? Just to kind of show you, and this is just information on 16S sequencing. Again, the idea is you PCR amplify the 16S ribosomal gene that all bacteria or most bacteria have this gene. It's highly conserved except in certain variable regions. And this is just a representation of that 16S gene, where the blue represents regions that are highly conserved. The red represents regions where you have what are called where you have more variability across species. And then usually what people will do is they'll sequence different sections of the 16S gene. Most commonly, people will sequence like V3 through V4 or just the V4 region, okay? But other people have sequenced all over. There's not a right way to do this or a best way to do this. Different microbes have different variable regions and different identifiability from these regions. But usually what people are focusing when they say we did 16S sequencing, they're usually doing some version of V3 or V4, okay? All right, that's usually what's happening, but it's not guaranteed. Now, interestingly enough, though, different species have different copy numbers of these 16S genes. And it turns out that that most bacteria, I've seen numbers between 80 to 85% of bacteria have multiple 16S genes, okay? And this actually creates a problem for some of the most common analytical methods that actually look at these 16S genes in isolation instead of looking at them in context. Okay, and if you just look at them in isolation, what you'll end up finding is that there's a lot of redundancies. And so if you maybe think of this as like one microbe, and then this is another microbe with different numbers and different locations of their 16S genes. But you might not be able to see this well, but I've specifically colored some of these to be the same color because it turns out that microbes could have multiple 16S genes, but from one microbe to the other, it could be sharing 16S genes, okay? And so that's why this becomes very difficult and tricky to analyze sometimes, okay? And so here's an example. In fact, Sean made this example, Sean Liu in my group. And if you this is just a toy example though, Sean, right? This isn't actually accurate. So it's just a cartoon example. Let's say, for example, E. coli had 4 16S genes. I think it probably has more than that. But you say, okay, here are the first 4 16S genes. And then if you have these other microbes that might be similar or related, they might share some of the same 16S genes. And so what you're saying is that what we're representing here is potentially you could have 16S genes 2, 3, 4, and 4 are shared across these different microbes. But what's unique maybe would be this first 16S gene that could be different. But then if you come in with sequencing reads, what you're going to find is that, okay, well, let's say the sample really did have E. coli and nothing else. But then I'm going to have sequencing reads that will align to this unique region, but then also to these shared regions. And then what it looks like is that maybe I do have Klebsiella here or Shigella here, in addition to my E. coli, because I'm hitting those 16S genes. And if you look at these 16S genes in isolation, it'll look like you have these other microbes, or you would have to maybe go to a higher taxonomy to be able to classify, well, I have this 16S3 gene, but it could come from any of those 3 microbes. Okay, to me, this seems obvious. And you might be saying, Evan, why are you talking about this? right? Obviously, the information that we have should come from this unique 16S gene. Let's rely on this because this tells me just by looking at that, that it looks like I have E. coli and not Klebsiella or Shigella because, why? Because I'm not hitting that unique region, okay? All right, that's obvious to me. Hopefully that's obvious to you, but everybody else who analyzes 16S data, it's not obvious to them for some reason because they don't take into account the context in the genomes, okay? Turns out that most analytical methods on this spectrum ignore the fact that most microbes have multiple 16S genes, and it creates problems, especially when you're trying to do species level analysis of 16S genes. In fact, for the last 15 years, people have gone in and said, hey, actually, it turns out you can't do species level analysis of 16S data. You're going to have to go to the genus level or higher in order to get useful information out of your 16S data. Now, if I was doing like an environmental sample, maybe it doesn't matter because a lot of times people are looking at phylum level or very high level taxonomies, because if you're looking at the soil here in New Jersey versus the soil in Utah, right, those microbes are going to be, well, actually, Utah's probably not going to have any microbes in their soil because they're dead, because it's so dry. But when you're looking at that, those differences will be very stark and very different. And you'll notice them very quickly. But when you're looking at a human, you actually do want to make very, very precise and be as precise as you possibly can. Because why? Well, it turns out that there are certain E. coli that are commensal and that they don't cause you problems, right? But some E. coli are very, very dangerous, right? And so we'd want to know which ones they are. Okay, and so again, in human studies, being able to go down as low down to species or even strain level analysis becomes a lot more important. Okay, and so that's one of the reasons why it was neglected, because a lot of these other methods were built in the context of environmental samples. Okay, all right, so these are just kind of, these next animations are just kind of illustrating this redundancy and this idea that if these reads get assigned to these 16S genes, we need to do what we call reassign them, okay, over or those mappings, basically say, hey, I don't believe those are real because I didn't get anything unique to that genome. Okay. All right. Again, seems obvious, but computationally, it turns out people are not doing this. So what we've done is we've developed our Pathoscope, and later on, I've got another slide of our Metascope tool. And what this is this is a plug-and-play software framework for analyzing metagenomics data. Okay, and so people will in the microbiome field, people will kind of separate 16S profiling versus shotgun metagenomics and metatranscriptomics data, and they analyze their data differently. And I'll tell you, I think that's a bad idea, because as it turns out, if you're trying to put things into context, it turns out that all you have to do is just align your sequencing reads back to whole genomes and then reassign anything that doesn't match up. Okay, and it turns out it doesn't matter if I'm doing 16S or shotgun or metatranscriptomics from my perspective. And this is how we've approached this, and it's actually led to significant increases and advancements in the ability to analyze these data. Now, here's an equation to scare you guys, but this is the magic secret sauce. It shouldn't scare you guys. This is an equation for a model that does exactly this. Okay, essentially, it's a mixture model that says, okay, well, I'm assigning these reads, I'm piling them up into different bins, right, or assigning them to different 16S genes, and then that mathematical model right there will essentially, as you iterate that model, will essentially say, okay, well, probabilistically, I don't believe that those reads should be assigned right there because I don't have unique stuff, so I'm just going to reassign them differently, okay? And so that's all the magic that's happening with that equation. It literally is doing exactly that, okay, from a mathematical standpoint. And the reason why we have to go mathematically instead of just something heuristically and obvious is that, well, what if these align probabilistically, meaning that maybe these align with sequencing errors or these align with sequencing errors? How can you manage that as well? And so that's why we go a little bit more mathematical, but that's what's happening here. Okay. And then we've developed this Metascope pipeline, which is specifically in R. Okay. All right. Now, cool. Interestingly enough, so Bree in my group, along with Tyler, he started this project. She actually analyzed 16S data using multiple different tools and multiple different databases. Okay. And what you're seeing is this in this figure, and I know there's a lot going on here. But what we had is a mock community example. So this is an example where they knew the microbial content. In fact, it was structured so that it was pretty even. So this is what we were hoping to see. Okay, so again, this is what this is this is supposed to be stacked bar charts. You've got samples on the rows, and then samples in the columns, and then microbes in the rows, and there are equal amount of microbes in every sample. Okay, so that's what this should look like. But then when you analyze it using different tools, so for example, using this Data2 tool, what you see is that it doesn't do a very good job at all at identifying the species in these samples. Okay, the gray represents proportions of reads that weren't properly assigned, or that weren't assigned to any microbe at all, whereas these colors represent reads that were assigned well. Okay, so some of these methods don't do very well. Some of them start to look like they're doing okay, right? Maybe that looks something like this. But if you can see, there's a lot of room for improvement in these algorithms. And some of these more common approaches, like this CHIME 2 approach or this Data2 approach, they're just not doing a very good job at identifying species in these samples. Okay? And again, the reason why is because they're looking at these 16S genes in isolation. And in this case, if you notice, 75% of my reads aren't going to get assigned to the right genome because they're getting assigned to multiple genomes. I don't know where they're actually from unless I take into account this one unique region. Okay, so that's what's happening here. And that's exactly why these methods are not working very well at the species level. Now, when you move to the genus level or the family level, it turns out that these plots start to look really, really good. And so existing methods do a great job at higher level taxonomies, but clear down to the species level, that doesn't work very well. Okay. All right. So we published this paper, and this was a very, very influential paper last year. Just kind of pointing out the fact that, hey, we're not doing a very good job. Now, these methods that we've developed, so this is our Pathoscope method, and then there's another method called Kraken, and these methods do take into account that structure, and they get us from about 30% accuracy to about 60% accuracy. Okay. Now, Sean, who's in the room, this guy's going to be famous, by the way. Sean has been looking at this problem of, can we do better at assigning species level, to making species level assignments. And what he's done is this. So you've got the ground truth. This is what we're hoping this should look like. Okay. Here's the most common method that everybody's using, gets about 30% accuracy. Here's our method that we developed. It gets us up to about 60-70% accurate. And then Sean, what he's doing is this. So what's happening is the following. The reason why these approaches aren't working is because they're looking at those individual 16S genes in isolation. The reason why these methods do better is they do take things into context. But the problem is that these algorithms that everybody's using use what's called a heuristic alignment, okay? Because it turns out if you're taking a sequencing read, let's just think of a simple example where you take a sequencing read, and maybe it's from a human, and you're trying to identify where in the human that sequencing read came from, okay? It turns out that the best way to do this would be to take that sequencing read and slide it along the human genome and see where it matches, okay? But computationally, that's intractable. you can't do analysis that way. Okay, so what people have developed are reference lookup tables or simplified approaches, where what you do is you take small pieces of the read and say, okay, where does that hit in the genome? And so you don't scan the whole sequencing read across the human genome. You just look for parts of the read in different locations in the genome. And it turns out those algorithms are what are called heuristic algorithms, don't guarantee that you're always going to get the best alignment. In fact, there's some limitations. And going back to potentially the 16S problem, what it means is that, let's say, for example, I had a sequencing read and that hit hundreds of genomes in my database. Think about this. If you're trying to even look at E. coli, well, it turns out that there are thousands, maybe even 10,000s now of genomes for E. coli, for different E. coli strains. And so again, I could have a sequencing read that's hitting 1,000 things in my database. Okay, and so some of these algorithms don't check for 1,000 hits. They'll just give you the best 100 hits and then return those alignments. Okay, and so what'll happen is that a lot of times these 16S genes occur in a lot of members in the family, and so sometimes you actually do miss the alignment that you're looking for. Okay, and so that's what's happening here in this case. This is why we're not perfect here. Our algorithm should get it, but the problem is that the redundancies lead us to sometimes miss the right microbe. Okay, so what Sean is actually doing to get himself all the way up to almost 90, I think, are you claiming 90% yet, Sean? Not quite? Okay, his goal is to get 90 though. He's going to be over 90. And we're not going to let him graduate until he does. Just kidding. But as it turns out, you can get up to we think 90 some odd percent if what you do is you add an additional alignment step. So meaning what we do is we do the analysis and get the best answer we possibly can, knowing we're going to miss a few things. But then once we're done with that, we're going to post-process those results one more time for things that may not look exactly right, and then do a better alignment. We can't do the best alignment for all of the sequencing reads, but we can take a subset and realign them and validate them that way. And so that's how we're getting this increased level of accuracy. And the great thing of it is with 16S data, we can actually assign species level assignments, okay? And it turns out that the field has believed you can't do that, but we actually can. And Sean's actually doing it. It's pretty cool, pretty exciting. All right, so again, like I said, he's going to be famous for this, I think. So you watch how this goes. All right. Questions or comments on this? I'm going to go into a little bit of the science now, at least non-computational science here. Okay, all right. So the other thing, by the way, is that we're developing dashboard and toolkits as well. These are analysis toolkits that are available in R that help to facilitate these kinds of analyses. So I won't say too much about this. Okay, now, before we go into TB, I want to talk a little bit about some of the work we've done in the past in asthma. And the reason why is I think it's quite illustrative on where we want to go with TB. Okay, so back in 2015, we were working with individuals at George Washington University, where, again, small sample size, okay? It turns out a lot of these are small sample size. And so there's a lot of work that still needs to be done in this space. But what we did is we took 8 individuals or 8 children with asthma, took nasal brushes from them, and looked at their microbiome. Now, these we're going to call them healthy asthmatics. Okay, so these kids are not sick other than the fact that they have asthma. And then we have some emergency room controls. And what we noticed is that there was in the nose of these asthmatic kids, these are inner city DC kids at Children's Hospital in Washington, DC, which might impact what we're seeing. But as it turns out, we saw a really interesting pattern that the microbiomes between these asthmatic kids and their controls are very different. The controls have, maybe call this a diverse microbiome, whereas asthmatic kids seem to have a lot more inflammatory microbes. Like, for example, this green represents a Moraxella cataralis, which is a commensal microbe. It should be there, but it shouldn't be there at that high abundance. And what's happened with these kids is their airway is exhibiting a very strong inflammatory phenotype. Okay. And then we actually see this through, and this is using RNA sequencing. In fact, this is RNA as well. But looking at RNA sequencing at this family of genes, these were genes identified to be associated with Moraxella infection in a mouse model. But if you look at it, they're just common. There's a lot of cytokines in here. There's IL-6, IL-8 is in here. So these are just inflammatory, NF-kappa B here, at least some components here. So you just think of these as inflammatory genes or immune-related genes. And what you'll notice is that these asthmatic kids have a lot higher expression in these cytokines and these inflammatory genes than the controls did. Okay, and interestingly enough, what you're seeing here is this is the signature strength of these genes. So the expression of these genes, kind of some composite expression of these genes. And this is the proportion of the microbiome that's dominated by Moraxella. And so if you notice that this inflammation signature tracks beautifully with the proportion of this inflammatory microbe as well. And there's this, except for this one child that didn't have, and this is this one right here, this individual didn't have Moraxella or a lot of Moraxella, but it had some other microbes that were inflammatory as well. So that's why you're seeing this inflammation signal. It's not really specific to Moraxella. Okay, so that's what you're actually seeing in this. And so this is promising, interesting data pointing out the fact that there's a strong connection between the microbes that are living in the airway and the gene expression and this is nasal gene expression, so that the host expression in the epithelium is interacting, or actually it's tracting well with the microbes that are actually present. Okay, all right, so that's what we're saying. I took this to our asthma docs. This is when I was at Boston University. I took it to some physicians that were working in there, and they said that they weren't interested in this. So we didn't do much with it. We picked a different disease. I guess we could do that. Interestingly enough, though, I lost that slide. Look, I'm missing a slide. Sean actually found a paper where people just last year were actually doing nasal microbiome profiling and actually using that as a biomarker for looking at exacerbations in asthmatic kids. And you can actually see from this data that this is not surprising, that when these kids are probably already have some sort of immune exhaustion going on right here, right? They're already inflamed. They're already fighting off just commensal microbes. And so then when they get sick with something, or when they go into an exacerbation, you can see that there may not be much left to fight off that exacerbation, right? So that's kind of the hypothesis that we came out of this analysis, and people are actually following that up now, almost 10 years later. So, okay, but we moved on. Oh yes, that slide's here. There it is. Oh, I have it blocked somehow. Okay, so this is that paper. Okay, people have actually did some nasal microbiome and COVID-19 as well. I'll kind of skip this, but just kind of pointing that using the nasal microbiome as a biomarker is very interesting and promising. Okay, all right. So as far as seminars go too, how much do these usually just go for an hour and then we're done? So I need to wrap up. Okay, awesome. Okay, so what I'm going to do is I'm just going to jump ahead just to kind of show you some of the work that we're actually doing, and I'll just give you a highlight on some of these results. Okay, so we've been working with our awesome team in Uganda, and we've actually now tried to study the nasal microbiome along with the host epithelial expression in the nose in individuals in Uganda, and we're looking at HIV positive individuals. So the goal is to look at individuals newly diagnosed HIV, okay, and who also have TB or don't have TB. Okay, so that's our population at this point. And then Katja's pictures here because she processed all our samples for us and is helping us analyze them. And then what Sean's doing is Sean's analyzing the data from a microbiome perspective. And what you actually see in this plot, it's not as clean as the other one. Here's the nasal microbiome from that small study. Okay, it turns out that we're not seeing as clean of results. Although, Sean, we should look at genus level plot on this one as well. I'm wondering if that'll clean some things up. Here's a species level profiling. There are a couple things that I do notice here in our TB samples, which I think are the ones here at the top. We do see a lot more Haemophilus showing up in a couple of these samples. But overall, it's pretty much a wash. There's not a lot that we're seeing in these microbiomes here. There is a little bit of a difference in our diversity of our microbiomes as well, but not something significant here. Sean was actually able to build some preliminary biomarkers using really to predict, can we predict TB using the microbes that are in the nose? So that's what he's been working on with some success, although this is just the training set, right? So he's not validating this on an independent test set. So this is kind of a cross validation on itself. So yes. So they're expert positive.
It's like, what is the bacterial load in these patients when you say TB positive?
When it comes to TB, I don't know if we have smear on these. Katya, do you know if we have smear? Okay, I don't know the answer to that. If you're looking at, you're talking about TB burden, right?
CTB.
Oh, why we didn't see TB? I think we did. Oh, yes. So we did see TB in some of them. Are we seeing TB in here? These are the most common species. There are some TB that we found in the nose. Okay, but interestingly enough, and this is where I'm headed with this, is that we were actually looking in the blood as well. Okay, and in the blood we actually did find TB. But interestingly enough, we actually found TB in both our TB positive and our controls. Okay, so this is something that was really, yes. So this is RNA. This is RNA. So we don't know if this is like free floating RNA or if it's RNA from actual viable bugs. We don't know the answer to that. But interestingly enough, in all of our samples, okay, even our control samples. Now, so these are individuals in Uganda with untreated HIV disease. Okay, so these are individuals who were we either grabbed into the control individuals or individuals that came in to the clinic and received their first HIV positive result. Okay, and then the TB were individuals who presented with TB, but in the process of diagnosing the TB, they also recognized that they were HIV positive as well. So these are the people that we're working with here. And so one hypothesis, and we need to follow up on this, one hypothesis here is that individuals with untreated HIV disease in Uganda might have extrapulmonary TB naturally, I think, in this population. Yes. So the fragments, I think we're looking at 150 base pair. I think they were paired end reads. And this is RNA sequencing as well. Now, interestingly enough, I think I've got this slide in here. Maybe I left it out here. Oh yes. It's right here. Let me grab this slide here. Here's a slide here that shows actually that we're actually getting full coverage of the TB genome. So these are different, these are these right here are different TB versions, and this is the coverage across those genomes. So we're actually getting full coverage of the TB genome from the reads that we're identifying in the blood. So this is kind of I don't know, a provocative question. So what we're trying to do is we're trying to validate this now in different ways. So this is unvalidated, but we do have TB, there are TB RNA, we know for a fact that there are TB RNA sequences in our sequencing data from all of these samples. Okay. So is it contamination? I sure hope not, but maybe if we want to get really provocative too, maybe untreated HIV disease. So that means your immune system is weak because of your untreated HIV disease. Are all of these individuals coming in Uganda? Are they all coming in with extrapulmonary TB? They've definitely all been exposed to TB just because of the prevalence of TB in Uganda. But we're finding RNA in their blood, TB RNA in their blood in all of our cases. And actually the ones that are actually sick, symptomatic, and expert positive are not showing much different in the amount of TB that we're finding in their blood. Interesting. So, and I don't have more than that.
Evan, how do you know that this is
Yeah,
RNA
and
DNA?
so we've actually done this. We've actually looked at this. We're not finding this result for example, in our Brazil samples. And although we don't have HIV positivity in that way, right? So it turns out if you remove the HIV positivity, this goes away. Okay, does that make sense? Or if you treat the HIV disease, this
Evan, how do you know this
so
is
the problem
RNA?
is finding the right validation set. Um, so yeah, got it. I agree. We haven't done it yet. So question, comments?
Do you have difference in the number of reads?
Like the read counts? I don't think the read counts are that much different. So we did RNA sequencing, and I think the total number of reads that we're targeting is somewhere between like 20 and 60 million reads. And then you do end up getting different amounts of microbe aligning reads in those RNA-seq data. But I don't think, Sean, can you confirm this? I don't think we have a strong difference in the number of reads in the TBs versus the controls. I don't think that we're seeing differences. So good comment. All right, I might just leave it at this. So clearly we've got to validate, we've got work to do. I'm not making any claims to nature. I'm not sending this off to nature anytime soon. But this is an interesting result that we can't explain. So we need to validate it and see if this really is true, or did something happen in our samples. And I'll leave it at that. Thank you, everybody. Yes.
You see much of a mix of age, sex, or nutrition?
Yeah, not in this study because we tried to match based on that. We focused just on adults, and I think we just have males here as well. We focused on adult males for this study. Would we see differences? Yes. So definitely age is a factor that drives the microbiome. Sex is a big deal actually for the TB disease so we would see differences if we looked at different ages and sex, but we didn't look at it here. Great question. Yes.
So for example, Alzheimer's patients, you have this beautiful correlation of post gene expression versus the.
Yeah.
So after treatment, do you see microbiome switch? So did you follow up with this study?
No, we didn't follow up with these. We do have other microbiome samples. Are you talking if you treat the TB or if you treat the Oh, right. So we are the answer is yes, we're working on that. The question is, have we done it? No. So maybe that's the easiest way. Yeah, how stable is it? Right. So once we treat these patients, does this change? Right. And part of the problem with these patients is that we're expecting this to be very messy data. And the reason why is because they're HIV positive as well, untreated HIV disease. And so there are studies out there that have looked at the microbiome of HIV, and clearly that treatment for HIV significantly changes the microbiome as well. And so we'd expect that would change significantly just by treating the HIV and then also treating the TB. There's a number of groups that have looked at the microbiome before and after TB treatment as well. So this is kind of the messiest, noisiest example you can think of is with untreated HIV, TB. So thanks, guys. Thank you, everybody. Appreciate it. Oh, David, did you have a question? I turned the volume down.
Yeah, can you hear me now? Can you hear me? Can you hear me? Okay, never mind. Never mind.
Okay, now speak.
Can you hear me?
I can hear you now.
Okay, great. So we're doing some work. It's still in the analytics phase with looking at cell-free DNA, amplification and sequencing. And, our unspiked plasma samples still have TB sequence signals in them. And
They still do.
yeah, so I think that's, I just guess that's that question. What can we do to do controls with this kind of study? But the other thing is that we're looking at DNA and you're looking at RNA, I guess. How do you know that this is not DNA sequences? You're looking cell-free DNA you're seeing instead of RNA.
I'm sorry. Yeah. What was the last part? You said,
How do
how
you
do I
know
know?
that this is? How do you know this is? How DNA free are your samples? How do you know that this is not cell-free DNA, not RNA you're looking at?
Yeah. So we don't know, right? And I guess the answer is we don't know. What the interesting point here is we are picking up this is an interesting thing is that we are picking up basically fragments from the whole genome. But this doesn't answer your question. This may be
Oh, it does in a sense, right? Because if you were picking up mostly RNA, wouldn't your 16S be? Wouldn't your read count in 16S sort of blow everything else out of the water?
Yeah. But so we're actually sequencing RNA here. So this is RNA-seq data.
Oh, okay, but then why isn't your 16S? Why don't you have one peak that's hugely higher than the rest with 16S versus?
Yeah, because we're not isolating 16S here. We're actually sequencing whole RNA. Total
Right,
RNA.
but isn't most RNA in TB?
Oh, I see. And so what you're saying, like for example, with this P. acnes here, right? We are seeing these spikes, which are probably the 16S genes.
Yeah, so why aren't you seeing that with TB?
Ribosomal. Yeah, we're not seeing them with TB. So I don't know. I don't know why. Maybe you're right. Maybe we're seeing something that's cell-free here.
Well, it's almost undoubtedly cell-free, I would think. The question is what is it? But I would suggest that this might be, of course, the scales are different. No, the scale's about the same. I would suggest this might actually be DNA that you're sequencing here. It'd be worth looking at the peaks. You have a few peaks, right?
Yeah.
But they're different at different positions also.
Yeah, with the different,
TBs.
and these are different strains, so maybe that's possible.
So I don't, if they're all aligned at 0, then they're not 16S. They're not, you know what I'm saying?
Yeah, right.
But I think that there's a lot of interesting things here between the clinical controls, between the laboratory controls, which get complicated for reasons I can't go over now. But when you barcode, your negative controls can be hidden, and whether it's DNA versus RNA that you're looking at here.
Right.
But it doesn't surprise me. And are we sure also this is TB? Or is there I assume you've used something like Kraken to look at other mycobacteria, and it's definitely
Yeah, so we've done, that's one thing because that's the one thing we know how to do, right? Is guarantee that these reads really are TB reads and not something else. And Sean spent a lot of time on this. We are very confident that there are TB reads in this data set.
Yeah.
So where they're coming from, we don't know, and why they're there, we don't know, but they definitely are there.
Okay. There's a bunch of technical things we're discovering when we do this, and where we can end up by having controls look cleaner than they really are, because when you barcode and sequence samples, you actually can't detect cross-contamination.
I thought there was some kind of email
that-
saying cancel.
That's Ruby. So she sent indeed out first. It was on, then she sent out.
I'm sorry, was that a comment made by Salvatore? No, it must be something else.
Yeah, it must have been.
Okay, we
something
should talk about this. It's fascinating though.
Yeah.
And these were all extracted and stuff in Africa, or was the plasma sent was your plasma sent here and sequenced, or?
Yeah, the blood samples were packaged in tubes that were sent here,
Okay.
processed here
Yeah.
in PedMini's group.
Yeah. Hopefully it didn't all just come from DNA floating around in the lab, but probably not.
Yeah, probably not.
But the issue is ones of controls. Very interesting.
Yeah.
Cool. Thank you.
We have to figure out we still haven't figured out what it is, so we'll have to keep working on it.
We should talk more about this.
Okay. Sounds good.
Are you going to be in New Jersey this week?
Yep, in fact I am. I just gave this presentation in person. I'm here in
That's
person.
fantastic. Okay, so I'll see you tomorrow. Okay.
Yeah,
great.
see you tomorrow on Friday. Okay, cool. Thanks, David.
Thanks.